Good afternoon, everyone. My name is Janice Nicholson, and I'm the Director of Communications for the Stem Cell Network. I wanted to thank everyone for joining us today for our first ever Early Career Investigator webinar series. We are excited to have you join us today and for the next 3 weeks, as this is a 4-part series. I'm going to kick things off right away by handing this right over to our moderator for today's discussion. Dr. Nicholas Dumont is an associate professor at the Department of Rehabilitation at the University of Montreal and a principal investigator at the CHU Saint-Justine Research Center. His research focus is on the understanding of muscular pathologies and their impacts on satellite cells, which is a population of adult muscle stem cells that's responsible for skeletal muscle regeneration. His recent work has demonstrated that satellite cells have cell autonomous defects in Duchenne muscular dystrophy. The objective is to define how extrinsic factors such as inflammation and fibrosis affect muscle regeneration in physiological and pathological conditions. Nicholas, I'm going to hand it over to you.
Okay, thanks a lot for the opportunity to moderate this session. I think it's going to be very interesting. So welcome to everybody to the first session of our first part webinar series designed to cover some of the main challenges and issues that early career investigators tend to face in their early stage of their career. So thanks a lot to the Stem Cell Network for developing and hosting this series. And a special thanks to the T-Cell Network for sponsoring this event. So the session today starts off at the beginning by covering key topics that are very important for establishing a lab. So in this session, we will cover topics such as recruitment and supervision of trainees, dealing with human resources, time managing, publications, and depending how much time we have left, we might also be able to talk about budgeting and mentorship. So we will try not to overlap with other sessions that are coming up. So as we said earlier, it's a 4-part webinar. So in the next 3 weeks, there are also going to be sessions talking about the art of funding your research, building your research reputation, and building national and international collaborations. So we'll try to avoid talking about these specific topics as they will be discussed in the next coming weeks. So there's a lot of challenges faced by ACI. I'm sure that we will not find universal answers to these specific problems, but we sure have a good panel of experienced PI that will help us to get good advice and maybe try to find common answers to some problems that ECI face. So today we have the chance to have 3 successful PI as a panelist. We have first Dr. Colin Krist. So Dr. Krist received his bachelor from the University of British Columbia, and he did his PhD at the University of Tokyo. He was a postdoctoral fellow at the lab of Margaret Buckingham at the Institut Pasteur in France. In 2012, he obtained a faculty appointment as an assistant professor at the Lady Davis Institute for Medical Research and the Department of Human Genetics at the McGill University. He holds the Marjorie and Gerald Bronfman Chair in Skeletal Muscle Stem Cell Research. Dr. Krist's lab studies the molecular biology underlying skeletal muscle development and regeneration, with current focus on translational control of muscle stem cells and myogenic progenitors. We also have Dr. Freda Miller on the panel today. Dr. Miller joined the University of British Columbia in 2020 and is a professor in the Michael Smith Laboratories and Department of Medical Genetics, and affiliate professor at the School of Biomedical Engineering and associate member of the ICORD. Prior to that, she was a senior scientist at the Hospital of Sick Children and a professor at the University of Toronto. She is a fellow of the Royal Society of Canada and of the American Association for Advancement of Science, and at the Hospital for Sick Children. She was Howard Hughes Medical Institute International Research Scholar, and the Canada Research Chair in Developmental Neurobiology. Dr. Miller is best known for work on dermal and neural stem cells and on mechanism that regulates neuronal survival and growth. And finally, we have Christina Nostro with us today. Dr. Nostro joined the McEwen Stem Cell Institute in September 2012 as a principal investigator. She currently holds the Harry Rosen Chair in Diabetes Regenerative Medicine Research. The objective of research is to differentiate pluripotent stem cells to pancreatic cells with the ultimate goal of generating functional insulin-producing beta cells for transplantation in patients with type 1 diabetes and eliminate the need of daily insulin injection. Dr. Nostro obtained a laurea in biology from the University of Florence in Italy in 2000. In her independent research career, Dr. Nostro has identified critical pathways leading to the efficient generation of HPSCs-derived pancreatic progenitors in vitro, and recently, through the use of proteomic approaches, she identified specific markers that allows the purification of these beta cell progenitors. Since 2015, Dr. Nostro has been leading a multi-investigator team aimed at developing novel transplantation approaches for type 1 diabetes therapy. So as you can see, we have a pretty good group of experienced and successful PIs. And I'm sure we'll have a very good discussion today. So in this first webinar, the first hour will be dedicated to a moderated panel discussion to get feedback of the panel members on their own experience and on their perspective on specific challenges faced by ECI. And in the second hour, it will be dedicated to question and answers from the audience. So please use the Q&A function if you have questions. I'll try to make sure to pass these questions up to the panel members. So if everything is okay, I think we will be good to go. The first thing I would maybe ask to the panel members, maybe just give general feedback of their own experience as an ECI, just to set how things went for them, and then we'll enter into specific topics. So let's start first with Dr. Miller.
Sure, and actually I'm glad we have a chance to introduce our own experience, because that probably leads me to some of the advice I think is most important for young investigators. And I was fortunate. I moved from San Diego up to Edmonton, University of Alberta, where I actually held my first position. And I was in the same position as all of you are in, which is that I didn't have equipment at the time. My lab was getting a few renovations. I didn't have a grant yet. But probably one of the most important things that I did is I was lucky enough that my department chair's lab was right next door, and I was doing a lot of molecular biology and biochemistry and tissue analysis. And basically, I invited myself to use a bench in that lab and to immediately start doing experiments. And that's one of my key pieces of advice, is that for probably at least a couple years, you are going to be the most highly skilled person you have in your lab. So if you want to keep your research rolling, which is really important, you need to basically act like you're still a postdoc in some ways. You're still in there, you're still doing experiments, you're still being productive. And that, honestly, that approach really stood me in good stead. The second thing that I did, and again, completely inadvertently, I would pass this on as also an excellent thing that happened, is I hired a technician right away. I didn't have a huge amount of money, but I had enough that I could afford that, as well as a couple of students. And the reason that was such an amazing decision, inadvertent decision, is because I had not appreciated until I opened my own laboratory how much administration was involved with setting up a lab. And again, to come back to this point that you as the principal investigator in your new lab are by far the most highly skilled person in terms of research. It's a waste for you to be spending 50% of your time doing things like learning your university's new financial administration system or things like that, which honestly can eat up a huge amount of time. So as I said, serendipitously simply acted that way when I started my lab. And it really, I think, allowed me to publish quickly because there was really not much of an interruption whatsoever in the research I was doing. And it allowed me to really not be too distracted by the other millions of things that you have to do in setting up a new lab. And I'm not going to go on and on because it was a very positive experience for me, very supportive. But I just like to say that this is closer to me in my heart than you might realize because I just moved to UBC and had to undergo exactly the same thing again, which is setting up a brand new laboratory. And even though I'm not as adept at the bench as I was when I started out of my postdoc, nonetheless, I'm very involved in the research, in this case, more computational things. And I have a fantastic lab technician who is helping me to navigate, I guess is the word, my way through all the administrative things we're now confronted with doing. So I think with that said, I'm going to pass it on because each of us, I think, are going to hear a different story from. Okay.
Thank you, Dr. Miller. Yeah, well, yeah, go ahead. You had something to
I was just going to pick somebody, but
okay. Well, I guess I'll have to pick. Dr. Nostro, you want to be next?
Sure, thank you, Nicolas. So I started my own lab in 2012, and I was actually lucky enough because I did my postdoctoral training in the same place where I started my own lab. So my training was done in Dr. Keller's lab, who's part of the Princess Margaret Cancer Research Center at UHN, and I was hired by Toronto General Research Institute, which is another institute under the umbrella of UHN. But effectively, I was located in the same tower. And that was a huge blessing because I definitely, I'm going to tell you what I've learned, and, hopefully, you can learn from my mistakes. I totally underestimated how long it would take to set up a lab from scratch because nothing was there. I had to essentially set up an entire tissue culture room for my lab, which meant ordering biosafety cabinets, and in the process, I realized that some of the, and incubators, and I realized that some of the things that I needed to have fitted in that particular room were not there. For example, a nitrogen line for the incubator. So there was work associated with establishing of, setting up of the tissue culture room that first of all, I had to pay for. and second, I had to wait for a contractor to come in and set everything up. So the entire process took, 4 months before I actually had a tissue culture room where I can work on. And as I said, I was lucky because my former PI was in the same place and I could actually do what Freda discussed. So I continued to do the work that I was doing as a postdoctoral fellow in the lab to make sure that, I wasn't wasting time. Because I was setting up the lab and it was taking so long, I kind of took my time in the hiring process. And this is something I also realized afterwards. I probably should have tried to set up the team as one of the first things, even if I didn't have a lab, because building your team is going to be crucial for you to move forward. You're going to train the people in the lab. And so if you wait too long, you're wasting valuable time. And if there's something that I could definitely would love to pass on is try to identify the people in the lab that you want as soon as possible. Like Freda, my first hire was actually as well a lab technician. She became immediately my lab manager. She was a very young person out of college. She didn't have master or a PhD, but she was driven, motivated, she was pleasant, and we worked really well together. So that was, what I needed for the beginning, because the administrative part, the ordering, takes a lot of time. Depending on the institute that you go to, you might be offered an assistant. I was lucky enough that UUHN offered this type of support, which was great because they obviously took care of all the HR part. But I know that there are situations like at the university where you wouldn't be most likely not having this type of support. And so in that case, probably the lab manager would be able to help. But, overall, I think the major things to, starting your own lab is making sure that you build your team as fast as possible and don't underestimate the amount of time that it would take for equipment to arrive in the lab. And so I guess I can stop now and we can continue the conversation on more about hiring.
Yeah, absolutely. So there's a lot to say. Dr. Krist, you want to follow up?
Sure. So good afternoon, everybody. If you can hear me okay. So my particular situation requires a bit of context. I grew up in Canada and I did my undergraduate degree at the University of British Columbia. But then I left the country in 1999 and I did my PhD in Japan and then my postdoctoral training in France. And that had some implications when I set up my lab because I was coming back to Canada in Montreal to a city and university where I had no prior experiences or interactions with a network of other researchers at the particular place I was, nor in Canada for that matter. And so that was one of the challenges that I had to overcome. And I would like to say that actually the stem cell network played a huge role in opening up a new network to me and getting me established within the network of stem cell research in Canada. And so if you are in a situation where you are also coming to Canada for the first time, I'd say don't hesitate to get in touch with the leadership at the Stem Cell Network and indicate to them how important it is to get a recommendation or to get a speaking slot, for example, at one of the meetings. And it's really important to get your face out there, your face behind the research as quickly as possible. And so that's one recommendation that I have for you. When I established my lab in 2012, I agree with everything that Freda and Cristina have said especially about doing research on your own for a little while, because you do have the best hands for research in your lab, and it's going to be like that for a good time. But I would also emphasize that it's really important to be patient when you are hiring people, and that you should wait until you get the absolute right person and the right fit for your lab. So patience, patience, patience. And so that has some implications for not only being the best scientist, bench scientist in your lab, but you may also have to go back to being the undergraduate student and stuffing your own pipette boxes for a little while until you get the right person in your lab. And in fact, that's what I did. I was quite patient, and I'm really glad in the end for my patience, because I ended up getting really good people to start off the lab. It did take 9 months for my first hire, but I'm the person who is my first hire is a research assistant and is still in the lab today. And she has been absolutely critical for the functioning of the lab as we've moved over the years. So those are my two biggest pieces of advice beyond what Freda and Cristina have already told you.
Thanks a lot for these first insights. Well, let's dig into how to recruit and train students. Seems to be something that's very important, obviously, for starting up a lab. We heard Colin talking about how important it is to recruit the good people right from the start. I would really like to have the opinion of the other panel members to see should we recruit as fast as possible, or wait and see and find the best person or the most talented person. How do we deal with starting the experience as soon as possible and then waiting for the good people to be in the lab? Do you have any comments on that?
Freda, do you want to go ahead?
You go ahead, Christina.
Well, I actually, I think I did exactly what Colin did, in a sense that I waited to find the right people, that I thought they were right for me anyway. But in retrospect, I think I waited a little bit too long, because I could have started faster. I was a little bit picky in my choices, and I think as a young investigator, you might not be under the radar so much that people applies to you. So you're actively searching for people, and it's coming from a big lab, you're used to a certain level. I don't want to say that it's but it's challenging. Let's face it. To find really, really good people is challenging. What I realized, though, is that being in a place like Toronto, you have access to a fantastic pool of student population. And you should be able to tap into that and find people that you can train. And if they're motivated, even if they don't have like all the experience in the world, you can definitely take them there with you. And I think this is, what I realized a little bit too late, but I encourage people to try and give a chance, to even undergrad or graduate students that you might not necessarily, on paper, give a chance to. Since
So I'd just like to pick up on what Christina said. And I think all of us, maybe not all of us, probably most of us though, when we start our lab, we somehow think that all the people who are going to work with us are just going to be like us. And the reality is that that's just not the case. There are a lot of amazing people out there who may have skills that you wish you had, but you don't have. And then there are going to be a whole other cohort that actually do have some of your skills. But as Christina said, they don't have the experience. Maybe they don't have the written proof that they're like that. So I would say that, to me, and I think this has held me in pretty good stead throughout my career, it's something that Christina said. You pick the people who have a good work ethic, who are really interested, and who are serious about doing it, that trumps all. You know what I mean? Everybody, in my experience, almost everybody who applies to your lab, first, they all have to have pretty good undergrad degrees or they wouldn't be applying, or even further training. So they're all at a certain level of intelligence. So that's not really the discriminating thing. The discriminating thing for success in people in your lab is personality, drive, openness to advice, to learning new things, to not being always successful and persevering. So those are the things you really need to, in my experience, to ask about when you're interviewing people. Because actually, this is no insult to all those people out there who maybe had 90% grade point averages, but in my experience, high undergrad degrees actually is almost counterindicative for coming into the lab, because what we managed to do in our undergrad system is select for people who are really good at memorizing things frequently and regurgitating them and getting a reward very quickly. You know what I mean? They work hard, they get the 90% in the class, and that's the reward. Whereas lab work is the absolute opposite of that. It's coming in, it's taking the small victories, your gel ran well, or you managed to subclone something, or your cells are healthy, and managing to take great pleasure in that and not being too disappointed when things don't work out the way you think they will. So anyway, I know that I'm talking at a level that's perhaps a little bit, higher than the advice that Christina gave you, which I thought was excellent. But I do think that you have to reevaluate who you think would fit in your lab, because it may not be what your preconceived notion of a successful individual would be. And that brings me, I'm going to be quiet in a minute, but that brings me to the other thing, which is that every lab culture is a reflection of the person who is the PI, which is you. And every lab culture is a little bit different. And so people who might work well with you may not be people who would work well with the supervisor of your postdoc lab, for example. And so I think the way you have to view it in a way is they're not really your family, but in a way it is like your extended family, your lab. And so you have to look for people who really you can communicate with and you can kind of work with, because you're going to be guiding them every step of the way. So I'm going to finish there. It's not really the practical nuts and bolts of when you hire people, but I think it's almost something that most of us never think about till we get into a situation where we've hired someone who really doesn't fit with us and it turns into a disaster. So I guess, Colin, why don't you take it over?
Yes, so of course I agree with everything that you have said, Freda, and emphasize that it's important for when you are hiring students, trainees, and research assistants that you need to consider their intelligence, their work ethic, motivation, all of that, and also that it's going to be that person will be a good fit in your lab. And what I want to say is that for all the members of this panel and the attendees, you're probably in your position because you are coming from a wonderful, wonderful lab that's been highly productive and working very well. And those labs that you are coming from have been able to recruit the creme de la creme of researchers, such as yourselves. And I want to emphasize that there is a much broader spectrum of people who are going to be applying to your lab. And so again, I just would emphasize that you should be patient about your first hire. And I know in my case, when I started my lab, I really felt pressure from myself that, okay, I have this lab, I need to fill these positions for it to be a lab. And I am glad in the end that I was patient in getting the right people into the lab and not just filling positions for filling positions. So be patient and wait for the right people to come along.
Christina, you wanted to add something?
Yeah, I just wanted to say I have maybe a practical comment, suggestions or something that I do that maybe other people would find useful when hiring students or postdoc. I know it seems a lot, but usually I also ask them to prepare a journal club, and I give them 3 options of papers that are in my field. And that gives me the opportunity to hear how they tackle a research project that is not necessarily most of the time in their field. And it also gives me an idea of how much they know about what we do. And it's very enlightening. It gives you an idea of like presentation styles and everything. And I found it, throughout very, very useful. So if anyone else wants to give it a go, I know it's asking a lot of the person you're interviewing because they're probably going to give you 2 seminars. But it's definitely been very useful and enlightening.
Christina, I'd second that. I think that's a really great idea when you're doing an interview and making your interview very vigorous. And that's a really good example of how you can do that to try to find the right people.
That was actually my next question. So it's partly answered, but I wanted to see if you have specific methods to try to find these good persons. So we heard about the journal club. Do you have specific technical questions for interviews? Do you have summer internship before they get recruited for grad student? Is there any other kind of methods that you use to find who's good and who's not?
Assuming you have maybe perhaps a few people in your lab already, it's really important that they talk to those people as well and that you get the feedback from those people, because this is again a somewhat cynical thing to say, but sometimes people can be very nice to you as the PI and then they'll be dismissive or not good to the people who work in your lab, or not as good. And personally, I think that's a disaster when you hire people that are kind of hierarchical like that, if you're trying to get a kind of a small cohesive group together. I also take reference letters extremely seriously, and sometimes you can't hold it against someone if no one knows them well enough to write a reference letter for science. But really, that's where you find out about work ethics, about their ability to get along with other people, something that you can't really judge in these more acute interview situations or even a journal club, right? And again, there's multiple ways things can go wrong with personnel. And if you get someone who's disruptive and or has bad characteristics, that's almost worse in a way than someone who doesn't have a great work ethic.
Thanks. We start to have quite a bit of questions in the Q&A. I'll maybe just start to pick a few that are related to what we're talking right now. There's one that's, do you have any advice about recruiting students for those of us at smaller institutions where the student pool is more limited than in major centers? So either that or specific places where you try to recruit students, or because it's definitely not easy for an CI to get noticed and that get a lot of good CVs. So any idea to suggest?
I guess it's also word of mouth. You reach out to your former mentor or other colleagues and let them know you're hiring, because they might be not in the position to hire, or other colleagues of yours might not be in the position to hire, but receive a lot of application. And if you let them know that you're interested, they could ask the applicants if it's okay for them to share their CV with someone else. And so I think that's very useful in a new place as well, especially if you don't have contact with the students, if you're not teaching and if you don't see them.
I think one of the beauties of the Canadian university system is that there are excellent students pretty much, especially at the undergrad level, spread out across the country in different institutions. And so, I can speak to, I know University of Alberta isn't a small university, but it's not as central, say, as McGill or University of Toronto. And there I found that by relying really on the local students rather than trying to recruit people in, I got some really fine people. And again, but I would like then to especially emphasize what Colin said, is if you're going to rely on that and you're at a smaller institution, then you really do need to just be somewhat selective about the characteristics of those people. That's all, but I think they're there.
In terms of recruiting students, I guess I would give advice also. You are at great universities here, and so I would give the advice to try to get some teaching at the undergraduate level as soon as possible. So they always, you hear a little bit about protecting your time for research and minimizing the amount of teaching that you do, which is important, but teaching at the undergraduate level will expose you to students, and students are always coming up to me after lectures to ask if there's positions. Almost every lecture that I've given, I've had one or two students come up to me afterwards asking if there's a position in the lab, either as a summer student or as a graduate student. So they are there and they are interested. So teaching helps a lot.
Thanks. So apart from recruiting the best students as possible, I guess the next thing is, how do you train them or give them your work ethic or workplace culture, or do you set the expectations right away? Do you have, or it's just in the air of the other how the other works, or you're in the lab to you micromanage? How does it work to set the workplace culture?
I'm going to start because I'm actually very guilty. I'm still working in the lab. I mean, I'm guilty. I love it. Don't get me wrong. I think it's great. And I hope that I'm not micromanaging. I think in the very beginning, I was there teaching, all the people that joined the lab in the first, 3 years, I would say I taught them myself. And it was a great, very rewarding experience, right? After that, I've relied on them and my lab manager to teach them, right? So I'm not teaching anymore, but I'm there occasionally now. And I find, the interaction, the fact that I'm there, it's fantastic because I can look under a microscope, we can discuss things in a very informal way. And but it is a bit unusual for someone at this stage to be still in the lab. I'm doing it, out of necessity. There is a COVID situation. We're working on shift, and I kind of picked up experiments again. But other than that, I wouldn't normally be there to run full experiments. But I could support people and help people, feeding things. I think for them it's nice to see me in the lab. It's kind of like you're leading a little bit by examples, and you share the same, issues and the success as well. You build, the team. So it has its perks, but it's certainly something that I don't think I can continue to do in the long term for sure. So at some point you have to really rely on the people that you hired to pass on the information and the culture as well.
So I think, I hate to be vague in my responses, but I really think that the way I manage, if you want to call it manage, the way I interact with the people in my lab, the students, is as individual as they are. And I think sometimes students come that have such just different personalities that I do a little bit of trial and error, right? I mean, some people do want you to, you ask, do you lay out the expectations? Some people need that guidance, right? They need to be told that, especially if they're younger, maybe they need to show up at core hours from 9 to 4 or 5, and they need to, when the technician's there and whatever, and when they can get lots of input. I have some students that I have to tell to quit working because left on their own, they just work all the time and burn themselves out. So I think, of course, there's a minimum expectation. And if someone isn't meeting that minimum expectation, and there are students that will start to come less and less, as I don't know if you've had that yet, Nicolas, but I think many of us have. Those ones you really do have to lay down the law. And then if it doesn't work, then you need to talk with them about why things aren't working out for them, right? And try and help them deal with it. So, but I think having said all of that, I think this is a much softer approach, if you will, than many other people have. And I just think that that's a reflection of what you view as the culture of your lab. And I'd much rather have people who are really self-motivated with guidance and help than I would to have people who are just doing, who are just there because I tell them they have to be there. So.
I can talk a little bit about personal experience. When I first started the lab, I think it's true that I was a bit of a micromanager. And I think that's within the context of starting your new lab. You need to pretty quickly generate 1 to 2 very good papers. And you, with a few number of students, not very many students who are trainees in the lab. And so for that reason alone, it's probably not the best idea to have, or the best timing for having students or other trainees taking off in different directions. That are activities that are really beneficial for larger labs. And so I think in the very beginning, you might not feel too guilty about micromanaging for that reason, because you do need to have some focus in your laboratory. And then as I'm progressing a little bit through my career, I can now start to be less of a micromanager, and I can tell you it's much more enjoyable way to do research is not to micromanage and to let the students have a little bit of leeway in their research. But I didn't think it was the best at the very beginning.
Thanks. We start to have quite a bit of questions on postdoc as well. Seems to the common things between 2 or 3 questions that I've seen is the difficulties of finding good postdocs. So, so did you have any issues in hiring good postdoc that usually try to go in bigger labs when you start because you're not well famous or at the beginning, or tricks to attract good applicants, where to find good postdocs. So is there websites that you recommend, or is it from your network, or how do you find good postdocs, basically?
I can start on this one because I just have a quick comment about the postdocs are generally looking to go to a big name lab, and that's important for a postdoc to go to a big name lab. And so you should be a little bit wary about postdocs that are coming to a lab as a early career investigator. And just to point out that sometimes the motivation to come do a postdoc isn't necessarily to come and do a postdoc. And that's especially true for international applicants, perhaps. So just be a little bit wary that you're getting the right person as a postdoctoral fellow. And I think that from personal experience, I've had a little bit more success with, no, I'm going to change that. I've had excellent postdocs in my lab, but I just want to say that they weren't necessarily motivated to be in my lab as a postdoctoral fellow. They were good researchers, but they weren't a postdoc. I don't know if that's not very clear, is it?
I think we get the point. Thanks.
I agree with Colin. I think it definitely improves with time, right? The caliber of the people that you are able to attract, because they also hear about you. In the beginning, if it's of help to anyone, I was trying to advertise as much as possible, in the UHN website or the general, through OIRM, SEMSA network, because you need to get the word out. I think now probably everybody could probably do it with Twitter. You make sure that you have a group of people that you connect with, and this way, I think the people who are interested in your work will come. And in the end, it's, it has to be a good fit.
Yeah, I guess I didn't actually worry very much about getting postdocs in my first 5 years. I had amazing students, and as we all know, people who spend their first 2 years just finding out how to work a pipettement and run a gel, by the time they're more senior, are really fantastic, and they're operating at the postdoc level. So that's more what I concentrated on. And even for the first 10 years, I was just really opportunistic. So there are people floating around who are really serious about science, but for personal reasons cannot move somewhere else, for example, or can only move somewhere close. And especially, I think, not in your first 5 years so much, but maybe the 5 to 10 year timeline. You can really get fantastic people then, but it's not by I never actively looked for them. I was just kind of had my ears out and kind of, let people know I'd love to have a postdoc, but because I think you already heard it from Colin and Christina that your lab probably is not going to be the first choice for a lot of people for quite a while, and but sometimes it will be because you're in the right place at the right time. So you got to look out for those people, and just not better to get really excellent graduate students in the really short term.
Thanks. We also have a question about international students. So does the panel have advice on how to evaluate international students for recruitment, where you know less well their academic system? Is it something that you had to deal with at the beginning? Did you recruit an international student? And if yes, how did you evaluate, especially if you cannot have them come for an internship or something like that? Is it something that you have faced, international students, or not especially?
These questions are making my lab sound like a disaster. I would advise, if possible, to recruit students from your Canadian or American institution. Sometimes an international student, depending on the country that they're coming from, will have a 4.0 GPA, and that 4.0 GPA does not translate to a strong GPA here in Canada. So you need to be aware of that particular issue.
I think, if I may, if you can find out someone who is from that location or university within your network, or not necessarily your network at the university, you can ask for their advice, because there might be like excellent people, and it's not giving them the chance just a priori. It's a little, I find it a little bit too harsh. I know it's hard because we all get millions of emails, I'm sure, that we can't judge. But if there is something that's in that letter, or kind of is special or makes you think, I would try to investigate a little bit more, because there might be a lot of talent, and it could be an amazing opportunity. Freda.
So this comes back to the reference letter I was talking about, and just what Christina just said. I have not taken that many international students, but the ones I've taken have done really, really well. But I had some kind of personal contact, whether I had met them at a meeting and seen their, if maybe they already had a master's equivalent or something, and they gave a great poster, and I had a chance to meet with them, or I knew someone who had actually trained them in some way in the country they came from. And but I agree completely that you need that. Otherwise, it's just it's sort of luck of the draw.
Absolutely. Have a personal connection with a student or somebody who knows the student when you are recruiting somebody from an international location. I think that would be key.
Thanks. So for the interest of time, I think we'll move on to the next topic. We might, if we still have time at the end, I'll come back with some of the questions that were asked in the Q&A regarding trainees. But the other thing I wanted to talk about is how to manage time efficiently. So maybe the first question, we're told a lot to say no to things, to do not get involved too much into committees and administrative duties and all these stuff. But these are also important for your CV, for your advancement, for peer reviews, and so how do you say no, and what do you say no to really keep your focus on research? Big question, I know.
Shall I start? So in the beginning, I didn't say no to anything. I said yes to everything. Which, maybe mistake, I don't know, but you feel like you can't pass on, committees or reviews, or, you're learning at every step, and you want your name to be out there, and you want to start collaboration. So I try to say yes to everything. It's more now that I'm being a little bit more selective, and choose really what I want to participate in. Because I think I've passed the 6-year reviews. I'm in a different stage. I really, those committees, those review, those panel discussion, conference, everything helps for your CV. And of course, it takes time for your research, but I think you learn how to balance it. So my suggestion is, if you can, try to participate and be collegial as well.
I also said yes frequently when I started my lab, and one of my major motivations to say yes as frequently as possible was, again, as I had said earlier, it was important for me to establish a network, and that was a really good way to establish a network, was by participating on student supervisory committees. Allowed me to get to know other researchers in Montreal very quickly, so that was very important. I also said yes to teaching. Again, this was also an underlying motivation to try to find undergraduate students that might be willing to come to my lab as a graduate student, and that was also very good. So I would say yes as often as possible, but be very strategic about what you're saying yes to. And then my other bit of advice, and it's important to be a little bit selfish when you are saying yes to people. So when you do say yes, you can also use that as an opportunity to get something in return. So if somebody's inviting you to be a supervisory committee meeting, you can say, oh, by the way, I would love to give a seminar, for example, at the institute. So try to get something in return to all these things that you're working on for the benefit of your lab. And then the last thing to say is stay away from administration. If the email asking for your participation starts with, please participate in our stakeholder advisory group, you should run for your life.
So those are excellent pieces of advice, because I think what I heard from both Christina and Colin is they said yes to everything, but what they really meant is they said yes to everything that had to do with science. So all of the invitations to review things, to give talks, to review papers, be on student supervisory committees, I couldn't agree more. But as Colin said, you really have to pick and choose. You have to do a few administrative things in your department, especially, unfortunately, I was looking at us as a group, and you're talking to a group of people here who are in research institutes, where they actually try to protect your time from administration. But if you're in a standard university department where you're already teaching like 2 full half courses or even more, you have the space to say no to a lot of university committees and things, at least for a couple of years till your lab gets going. And I would encourage you to take advantage of that and instead be on student supervisory committees or review a paper or whatever.
Thanks a lot for these very practical advices. Also a follow-up question on that from the Q&A. How do you deal with all the emails? So do you have some specific methods not to waste too much time on the emails?
I can give a little bit of feedback. One of the things that I've done recently is use some other technologies to communicate with my team. And so one app, for example, that I'm using now is called Slack, which is basically a messaging service that you can use to share messages and materials with your team. And that's beneficial 2 ways. One is emails are kind of cumbersome and you still feel motivated to write a bit of a longer message, whereas a chat is, you can write a chat in 3 words and get what you want. So nobody has the expectations of the formalities that come with email. And the other reason that has helped is that by focusing on what's happening in Slack, I focus on what's important to me, and that's what's going on in my lab. And it has trained me, just by having that option available, it has trained me to stop looking at email other than once or twice a day, maybe at the beginning or at the end of the day. And then I also set up a lot of filters so that a lot of the administrative keywords like stakeholder advisory group, that immediately goes into a folder that I try not to look at all.
Yeah, just to echo that I actually have a couple of email addresses, and one of them I share with my things like my lab ongoing research, which is kind of the equivalent of your Slack channel. And so I pay attention to those things pretty quickly, but otherwise unless it's something really important to me that I need an answer right away, I'm pretty negligent in my emails on a daily basis. Every day I do them, but I do them at 1, maybe 2 times. And it's amazing how much time that frees up.
I'm actually learning a few tricks here. I think I'm gonna take you on the different type of email for the lab, because it seems like, you know, a good way to filter. And for the most part, I just limit the time. So usually I do the emails like early in the morning, but then anything that comes like later in the afternoon, unless it's, you know, I give it a quick check, but unless it's something that I know it's important, I don't, I try not to, you know, try to stay focused on the work in front of me. It's very distracting otherwise.
Thanks a lot. So we have another question related to managing time, but on a different aspect. Here is any tricks on maintaining a healthy work-family balance when you're starting up a lab and have young kids?
So I'd like to speak to that because I had my first child while I was right before I started my postdoc, and my second one within a year of starting my first job. So I had that particular issue in spades, if you will. And I made a decision when I opened my own lab that I was going to do it to the best of my ability, the way that worked for my personal situation. And if that wasn't good enough, then, well, so be it, right? I mean, because my family was very important to me. And what that really meant in practical terms is I worked a certain amount of time and then I spent a lot of time with my family. And it worked. You know what I mean? I was super efficient when I was working, but I mean, that was it otherwise. And the other thing I did is throughout my career, give yourself permission. Well, we've all had permission for during the pandemic, but give yourself permission if it works for your family life to do most of your writing at home. Because when you have young children, it gives you a chance to be at home with them. If they're taking a nap, you could work, for example. Don't let anyone guilt you into feeling like if you're not actually on site in your office, that you're not really working. Now, that may resonate differently now and after the pandemic, post-pandemic, but I think before the pandemic, that was important.
Thank you so much for that, Freda. It's so refreshing to hear because, it's easy to get guilty into, working in the lab or in the office all the time. I'm guilty of that. And, things will definitely change now. We've realized that we can do this better. And it's up to us to show this also to our trainees and give them the opportunity to take time and spend time with their families.
And yes, I agree as well. I think keep in mind that science and academic life can be a tough call, and your family really helps you get some perspective on life about what's important. I don't think in my professional experience, I haven't had any issues trying to balance work and home life. I think I've found a very good balance, and I really appreciate it. So I think you should do the same.
Yeah, if I can add something to that as a personal experience, I personally decided with my kids that I would not let them know that I'm working. So if I have to work on weekends, I'll do it when they nap or when they're asleep, or so that their priority is my kids. So I won't, and then after it's work. So I try to manage my schedule so that they did not even notice that I work during the day if I had to during weekends, for example. So I just try to set that priority first, and then move the rest accordingly. Another, I think, important things in that I would love to hear you talk about is how do you manage lab meeting efficiently? So what do you do in lab meetings? How frequently? Do you have any tricks to share? Freda?
Okay. I try to have weekly lab meetings even through the pandemic. And it's not great on Zoom, I have to be frank, because people are very inhibited about speaking up once you're in a group of more than 4 people or so. Those are our lab meetings are somewhat formal in the sense that we do lab updates at the beginning, like critical things like that. The centrifuge that everybody relies on has quit working as of yesterday or something like that. And then 2 people give presentations, and it's their choice what kind of presentation they want to give. If they're preparing for a job interview or postdoc talk or thesis defense, they give a formal one, but otherwise they can do a journal club or they can present their latest research. But in addition to that, I also do smaller group meetings because, in particular in this age of Zoom, because what happens in the big ones now, and even to be honest, in person frequently happens, is you get a few dominant people who talk and ask questions all the time, and then you get the quieter ones that just sit there. And so in order to enable the quieter ones to have a chance to practice participating in a really full way in community science discussions, I try to have these smaller group meetings as well on a needs basis.
In my case, I have individual meetings with my trainees once a week, which I think is very important, and that's a time to discuss informally about the science. And so we just look at the lab notebooks together and the data together. And then I have more formal lab meetings with my group where everybody is involved in giving presentations, and we're also doing that currently by Zoom. And I guess for ECIs, I'd recommend in that regard to just be a little bit flexible about the timing in between lab meetings, because you do have a small group, so it doesn't necessarily make sense to have a formal lab meeting every week, expecting your trainees to present at them. So you need to space those formal lab meetings out a little bit, and then maybe keep the informal meetings with your trainees much more frequently and be rigorous about it.
I'm following the same methods as, a weekly lab meeting. The lab is big enough now that it's about 8 people that present on a weekly basis. So it gives enough time for them to present their latest results. It's formal, but they're presenting the raw data, so we can all pitch in and discuss. I have individual meetings on request, let's say. They're not set, so when anyone wants to talk to me, now we'll just set up a time on Zoom or Teams.
Great. That brings us maybe to the next topic I wanted to talk about, which I think is very important obviously for everyone, about publications. How to maybe what was your strategy regarding publications? So when you started, did you aim to publish something quickly, even if it was not that big, or did you go all in and try to go for the big publication right away? Just have your impressions on what's the best strategy. What did you do when you started your lab?
I'm going to speak now. I don't think it's fair for me to speak about when I started my lab, because the publishing landscape has changed so much since then. I mean, I just went for productivity. You know what I mean? Publish really beautiful papers as quickly as I could. I'm going to say 2 more generic things, though. One is that what really matters isn't the journal that it gets into. What really matters is how beautiful it is. So you know what, to be this fixation on getting it in a trendy journal, yeah, I mean, maybe it's gratifying, maybe it gets noticed a bit more, but the bottom line is you're writing papers first because that's what we do as our business, but secondly, because we need to renew our grants and things. And I can tell you that to a grants panel, what matters is when they look at that appended paper, that it's really quality work and it really says something. The second thing I'm going to do is wear my hat as someone who has sat on promotions committees at all the institute, at both McGill and Toronto. And I can't emphasize enough how important it is to prove that in your first 5 years you can be productive. Really, that is probably the fact. You need to be able to get a national grant on every university I know about, and you need to be able to show that you can publish papers on your own. You can still have, many people still have, just because of the nature of publishing now, still have, maybe their ex-lab, there's a few people from it that are in the middle of the work. That's perfectly fine. That's not the issue. But it has to be clear that it has come from something that has been predominantly done in your lab. And it should be preferably more than 1 paper. So if you can still go for the big Nature paper while you're doing that, good luck. But given how long it takes to publish these days, I don't think that's necessarily the right strategy.
When I started my lab, we were fortunate enough to have started immediately on a really nice story. And after 4 years of research, I had the option of either going for a very big impact paper or splitting that paper into 3 individual papers of potentially lower impact. And I decided to go for the big impact paper, and that had a lot of headaches that go along with making these big impact papers. But there's an opportunity there, and I seized it, and I'm really glad that I did. And it definitely helped on my tenure application, having that one paper along with 2 smaller papers. It was that part of my productivity was considered a home run at tenure. So.
But Colin, can I say something?
Yes.
You had 2 other papers.
Yes, and they were.
So that's not, I think that that's perfectly legitimate. As a matter of fact, I would encourage people to be productive with bread and butter stuff and going for the big, you know, hit research, right? I think that's a good strategy. I think though frequently what we're talking about is no papers.
Yeah.
And going for the one hit, right? Which is a very different thing.
Yeah, it can be a rabbit hole, can't it? Because that you can put a lot of time into that one hit wonder, and it doesn't actually end up getting published where you want it to be. So you end up spending a lot of time. So
Yeah,
be careful.
I agree. I think in the end, you wanna, as Freda said, you wanna create something that you're proud of, that your peers would recognize, and even if it's not in Nature, it gets referenced a lot because, you know, other people see the, you know, the novelty of your work. So I think that speaks a lot. And try, you know, if you can, to get advice from, especially in the beginning, I would say advice from collaborators or mentors and see what they suggest, because they've been through that and they can tell you, okay, you know, you don't have to listen, but, you know, hearing different voices can help you decide, I think, in the beginning and see whether there is really the potential for a big publication or not.
And on your personal experience, so maybe on your experience as member of committees or panels, what would be a reasonable time frame for a first publication? So should you aim to publish in the first 1 year, 2 years, 3 years, or just before your tenure at 5 years? Does it matter? So how fast and what you should aim for a first publication?
I mean, in terms of getting research funding, for example, when I got my very first CIHR grant, reviewers of my CIHR application, I think I got mine 2 years after I started my position. And there wasn't one reviewer who insisted that I needed a publication of my own at that time. And I think you can give yourself up to 3 years before you need to start showing some productivity. I think. Freda, do you agree? Or?
Yeah, I agree, because usually at most universities at 3 years, there's kind of a check-in point, right? I mean, they call it probation, but it's not really probation because no one's going to get rid of you then. But it would be really optimal to have one paper by the probation time, by the time you're putting that little 3-year package in, if you could, right? Even in revision, right? Even in revision, because then you can show it to them and they can see you've done a beautiful job and it's really something real. So, Cristina, was that what you aimed for, or?
Yeah, I mean, I don't think you can plan these things very well, but ideally, yeah, that's what we try to strive. I think it took us, the very first paper came out after 3 years, but it was still with my former PI, and the one fully independent came at the 5th year, actually.
But with your former PI, who was senior author, Cristina?
He was. I was the corresponding author, but I was still first. I was the first author corresponding.
But you were corresponding.
Yes.
Okay, because that I think
that made the difference. I know, but he was still there, like in last name.
Yeah. Okay.
It was definitely recognized that the work, you know, it was a lot of work that was done in my lab, but it started in Gordon's lab. So I think, you know, to be fair.
So I'm going to say something that may be somewhat contentious with, more senior people. But I think you should, if you're in the situation Cristina just described, first, I think if it was done in that lab as a postdoc, then of course those people should be offered authorship. But I think you might want to push for being the senior author. You might want to say, hey, you know, you're a well-established person, and you can even be, because what I frequently do is put myself second last
Yes.
and let them. Anyway, I'm not, Christina, you don't have to go
No,
into
no,
the
no.
details.
I totally agree. I think it's something that at some point, you know, especially if you reach a certain level in your career, it's nice to offer to your trainees.
Yeah. But don't wait for the offer, is what I'm saying.
If you don't ask, you don't get. That's very important.
And you know, I don't know about you, Christina and Colin, but that was a huge lesson for me, that I had to ask for things, because I kind of was very idealistic, and I thought if I do really well, people are all going to do the right thing, right? Give me a raise, or you know, offer me senior authorship or whatever. And actually, it's not that people aren't good people, it's just they frequently don't even think about it.
Mm-hmm.
Right. And so I think that's a big message, is ask for what you need, because don't just hope that it's going to happen.
Very true.
Eric Shoebridge, who is the chair of our Department of Human Genetics at McGill University, gave me some very good advice one day. He told me also, if you don't complain, we don't know that you're not happy. So it's important to speak out about the issues that are bothering you.
Thanks. And still about publications, do you think it's a good idea to publish protocols or reviews, or just to have your name out there, or maybe help the productivity of your students at the beginning when they're applying for fellowships or something like that?
So I thought about this question before we had this meeting about what I would say. And I would say, sure, it's fine to publish things like that, but I would make sure that you have your priorities straight. So the priority is reviewed papers. The second priority is probably grants. And then after that, if you're sitting around and you know what I mean, you got some spare time and you're not worried about your home life, then go ahead and write a review or a protocol.
I agree. I mean, in a ways, I find it's nice for students or postdocs to get involved, because they really delve into that literature, but it takes so much time, so much time. So be strategic for sure.
Another question that is kind of new in the field now is, do you publish on pre-publication websites, bioRxiv or others, especially as a young investigator, when your reputation is not established yet, and you don't want your ideas to be stolen, but you want to have your kind of your niche where you're already established. So you have any idea about that on a ECI perspective?
I started posting my publications to bioRxiv prior to as a preprint. I wish it didn't, but it does cause me a little bit more stress for the reason that you do start to worry about having that out there and the amount of time that it actually takes to get it published. But in retrospect, I wish I had not been stressed about it, because there was no issue that I've experienced so far, and I will continue to publish at preprint servers like bioRxiv in the future. I think it's helpful. Also, I have also started putting bioRxiv preprints on my CV and on CIHR applications, for example. I don't know if there's currently any regulations about that, but it helps to show some productivity as an ECI if you have a manuscript in preparation that you can show.
Just with regard to that, actually, Colin, I think you can, for many journals, you can quote preprint like that in the reference list, whereas you cannot, without great difficulty, put the personal communication thing in. So, yeah. Well, okay, since I'm talking anyway, this isn't fair. I'm the oldest person in this conversation, so probably the last to adopt the preprint. And there's no real reason. I've kind of been leaving it up to the people in my lab, whether they want to go that way or not. And none of them have felt strongly, but I really should start doing it for all the reasons Colin said. And honestly, in terms of being scooped, my entire career, there's only one time when someone did exactly the same study we did and we ended up not being able to publish. So, you know, I mean, you can worry, but as Colin said, I don't think frequently it doesn't happen.
We haven't done it yet, but we're planning to do it. And we have a paper now we're trying to resubmit. I believe we can probably go in bioRxiv with the original submission, not, of course, not without the revision, but it's the plan. I think we have to move forward with time.
Another advantage these days about presenting your research either by a talk or by pre-publication, prior to publication, is that instead of thinking about getting scooped, you can think about it in terms of setting your mark, that your field knows that you're working on this project. And I think, we're in 2021 now, and the days of people viciously trying to scoop other people's research I hope are over. I have not experienced it myself, and I don't really consider it a big issue anymore.
I agree. It's certainly, it's out there, right? That you're doing the work.
And maybe in line with this, would you recommend to enter dialogue with the competitor, somebody who knows that is working on this or have interest in your field, either at conference or even just sending an email or giving a phone call regarding the publications that you're about to submit? Or do you see any advantages or inconvenience or risk to this kind of
We've actually, I'm thinking of one particular example where one of my colleagues in the field found out we were working on the same thing, and we ended up publishing back to back in the same journal. And so I think that's the more common experience, actually, Nicholas, than the adverse experience. And I really believe that our job as scientists is to push science forward, and that the more open we are in communication, then the faster that's going to happen, which is the other reason for putting things on the preprint service, because it takes forever to get papers published now, right? So you're sharing the science so people can, instead of repeating what you do, they can go to the next step.
Everybody agree on that? Good. So good. We're still in time. There's also something I wanted to talk about with so first for the audience, if you have questions regarding these things, feel free to put the questions online on the Q&A. I can follow up on that. But in the meantime, I want to also talk about budgeting, not on the grant writing part of it, because that will be covered in next webinar, but just how did you manage your budget when you started? So with your startup, did you invest and go all in with what you have, or do you keep it as a kind of safety net in case something goes wrong? Do you have any suggestion on how to use your startup?
Okay, I'm saying a lot of sort of maybe inflammatory things, but I'm going to say it now, which is that I honestly took the attitude that if I produce good work, that the money would come, and I really was not cautious whatsoever in my budgeting. I just, if I got a great student or something, I just hired them. And I always had the attitude, you have to do the best experiment. And if it costs more money to do the best experiment, you just do it, right? And you know what? That worked. But I'm not saying it's going to work for everybody, but the problem is if you're too conservative, you don't get the momentum, right? And as a young investigator, you need to get the wheel rolling, so.
I agree with Freda, actually. I think you shouldn't worry about the money in the beginning, just go for it. So if you can produce with what you have, then what's the point? You know, then you have to figure out something else later on. So just go for it. And what I try to do is invest a lot in writing fellowship and scholarship for my students. So I dedicated a lot of time there because it can save you a lot of money. And it's, you know, I think in Canada it's relatively easy compared to other countries to secure funding for salaries for your trainees. So that's the only way that I try to budget. Other than that, it's just, you know, not be wasteful, but, you know, if we want to do something, we order it, the antibody, whatever it is, you know.
Yes, I already agreed with what's already been said. Go for it. Use your startup funds to the best of your ability and do good research with them. I feel our startup funds are pretty generous in Canada, and it lets you get a really good start. Nico, I have a really good story to tell about budgets for equipment. I don't know if that's part of this talk or not.
Yeah, sure.
It's just a story. When I first started our lab, we got a CFI allocation to purchase equipment and to start the lab up. And so this is an example of, I know how to do science, but I don't know how to make a budget or get quotations. And so when I was doing this assignment for the very first time, I decided I'd get all of my equipment mainly from one vendor, or the majority of my equipment from one vendor, and it was about 80% of all the equipment that I had was from one vendor. And they gave me all these quotations and I submitted my budget. And it was just fortuitous that our finance office at our institute looked at the budget, and he called me up on the telephone and he said, you know, this is crazy, you're getting fleeced by the vendor. And he personally called the sales rep of this one vendor and said, it took him about 20 seconds, I want you to reduce this overall budget by 40%. And they did it. And they did it. So 40%, or virtually about 30% in total of my CFI budget, I had more money to spend on equipment. And I was a little bit miffed that I was getting fleeced by this vendor. And so it kind of resulted in kind of a bad relationship with them over term. But I'm really glad that our finance person had flagged that and had helped me out. So I would advise you guys to become friendly with the people in finance and have them look at your budgets and your quotations. Saved me a lot of money.
Yeah, for example, here they will definitely, for a bigger equipment, they will ask for bidding. So you wouldn't be allowed to go for one vendor alone without having different quotations. So there is a system in place to make sure that you're kind of protected.
Okay, I have a question related to budget. Due to the COVID-19 situation, some reagents and consumables have doubled and tripled in price in the last year, making it very hard to stay within the budget. I realize that we probably all struggle with this right now, but I guess with some more experience as a PI, you might have some suggestion to how to navigate this. So I guess, Colin, you give a bit of advice on this to have people in the finance help you out, or you contact the rep, I guess, to get some good deals that are not offered right away.
I don't have personal experience to draw back on this one. So I mean, the short answer is no. I see Christina for hand up, so I'm let her.
Yeah.
I would suggest if there are reagents that you're buying and you know someone else in your department is buying the same thing, try to team up with them and ask for larger quotes. You might be able to get bigger discounts as with larger amounts. This is normally what we do with cytokines. We buy large amounts rather than small amounts to get the best quotes possible. And try to have a tough negotiator in your team. This is my lab manager. She deals with the sales reps, and she has a great relationship with them, but she's definitely always on email or call to make sure we negotiate the best price.
Thanks. I have another question that's bring back more to trainees, but I think it's very relevant for us right now. Any suggestions to ECIs for how to motivate their trainees and keep the research momentum during the COVID-19 pandemic? So how did you deal with the current situation and have people still motivated to do their job?
Well, I think the first thing, and I think probably all of us have thought a lot about this, is to be really kind to them, because, it's bad enough for us and we've got jobs and we've got our futures, at least for the next while, pretty well worked out. A lot of them are living in frequently by themselves in not very nice little spaces. And I think you need to be talking to them a lot. And it's, to go back to this thing of each one of them is individual. I have some people in my lab who actually, one person told me they like it better. They're perfectly happy to sit at home and do computational stuff all day by themselves. But then I have others who are really suffering, and all you can do is be excited about the data they do get. Do you know what I mean? So they can actually, if they talk to you, you can be their cheerleader, even if you feel crummy yourself, and even if you're suffering, you have to be that. And I can't think of any other way, actually. I also changed the nature of people's projects. So because, well, like a lot of labs, we're doing more and more big data analysis. And because of that, right at the start, I shifted most people to having a project that was half computational, half bench. And so they could, even if they ended up not being able to go in as much as they wanted, they still had something they could work on and feel like they had achieved something with. I don't know what else to say, because there really isn't one size fits all right now.
I'm fortunate at the moment because I have some trainees in the labs who are have maintained their motivation and they're working quite hard during the pandemic. And one of them said to me that, it's difficult during the pandemic to do research, but on the other hand, I feel that I have a very safe environment within academia to come and work, and I'm very grateful to be able to come to the lab when many people are obliged to stay at home. So I think you can feel a little bit proud if you can set up an environment for people to feel safe during a pandemic to do research, and then I wouldn't go too far over that in terms of trying to demand data from my trainees during this time. Just not, and as Freda said, just be very kind to your trainees. Some of these are international students that have no network here in Canada and are spending a lot of time at home and are just grateful to be able to come to the lab and do work. So that's good. And then the other thing to say was about research personnel in your lab. We've hired research assistants and whatnot. And there was a time during the very first wave of the pandemic when there was talk about letting people go or laying them off because they are obliged to work from home. I did not do that. I maintained all of the people in my lab who were employees, even if they were obliged to work from home, I maintained them as a full-time staff. And I'm 100% grateful that I made that decision and did not go the other way. So be good to your people and they'll be good to you.
Okay, thanks. So maybe the last topic I wanted to cover for today's webinar is about mentorship. Not especially how to be a mentor for the trainees, but how to interact with your own mentor. So having a scientific mentor, how do you choose it? And especially how do you take the best of that person? So basically, how was your experience with your mentor, and do you have any advice to share on how to take the best out of it?
Maybe I can start. I was assigned mentors both at the institute and the university, and of course, you meet with these people, but you should also find informal mentors. I find that these are actually people that you can go to on a much more regular basis and discuss, your grant or your ideas, and bounce off ideas, even in front of a beer. So it's a colleague, who has the function of a mentor, and it doesn't have to be limited to one. You can have different people filling different areas of science, and I found that to be super useful. And also, they don't have to be in the same location. It can be a different university, a different country. So I think find your informal mentors, because you might be able to relate with them, in a different way.
Yeah, Christina and I have had the same experience. I think my informal mentors have been much more valuable to me than a formal mentor that has been assigned to me.
Yeah, and as someone who I've still fortunately had mentors throughout my whole career, but who's frequently in the position of being both an informal and a formal mentor. It's kind of like with collaborations, right? Things can look great on paper for a collaboration or for a formal mentorship relationship, but actually maybe the people don't hit it off quite as in that very personal way. So I think mentorships work best when actually the person is almost your friend, basically. So, and that's where the informal mentorship thing comes in, right? Find people you're simpatico with and who you respect, and who are they could even be at the same stage of career, but frequently they're more senior, and just listen to them, and don't feel guilty if you're not seeing the person who is formally assigned to you as a mentor very often, just because perhaps it's not as useful a relationship. But on the other hand, don't hesitate to reach out and ask them for help, because that's the other thing that happened to me frequently. Was someone would be assigned to me and I'd be their formal mentor, and I would just never hear from them. So, I would be happy to help them, but unless someone reaches out, how can you help them? So
I guess that brings back to the thing we said earlier about if you don't ask, then nobody will know if you have any trouble. From that perspective, would you ask your mentor, formal or informal, to invite you for talks, or to be collaborator on his grant, or to co-supervise students, or things that will help to get visibility as a ECI? Do you go and ask for it?
I mean, if there is ground for a collaboration, sure, I would have no issue whatsoever. And talks, if it's a different university, why not? I mean, if I have a story and I feel, it could be of interest, yeah, I think so.
I had the experience again because when I first arrived back in Canada, again, it was because I didn't have my network, and where an informal mentor that I had, I just approached them and said, I want to be able to do this type of experiment. I don't have the expertise. Can you help me, introduce me to somebody who does this? And he got on the phone right there, and within a minute he had me a new collaboration for a person with the expertise. So these are ways where mentors can help you.
And I just like to say that a good mentor, some of the very best mentors I've had, they were just really thoughtful people who would get me invited to speak at a conference, or, you know what I mean? But I mean, that's somewhat more of a rare thing, but there were several people through my career who really helped me a lot in that regard.
Thanks. So that covered the mentorship aspect also that I wanted to talk about. I'll maybe just we have a couple of minutes left. I'll go back to some questions that we have. I think there was especially a lot of questions on trainees and supervision. And one, I think, aspect we did not cover was a question that I have here is how to deal with a situation when you've hired a person and they end up not being the right fit. So how do you deal with people when things go kind of wrong?
Just be patient and don't do it.
I never had to fire anyone. But I've heard stories, right? And I think if things don't work out, you probably have to have a report of why things are not going well. If you need to have the facts, right, in front of you to go to that, make that decision. I think if it doesn't work out, it probably doesn't work out both ways, right? It's not just you. So I think in the interest of everybody's sake, you might have to, let someone go. But it's probably not an easy process. I think if it's a new hire, you may have 6 months, sorry, freedom to interrupt, of a probation time. But after that time,
Yeah. I just wanted to pick up, Christina, on something you just said, right? Which is that frequently if something isn't working out, you have that student who just starts coming in 1 hour a day or they just clearly are unenthusiastic, whatever. You know what? I've had good success in just sitting down and talking to them and basically, why are you only coming in? Are you just not, maybe science isn't for you, or maybe this lab isn't for you, because obviously, as you said, Christina, if it's a two-way street, unless they're completely delusional, they're going to know that things aren't working out right. And if you can convince them that perhaps they have maybe they're great communicators, or maybe they would be fantastic doing something else, you can guide them that way. And I have had people leave the lab with that kind of a conversation. Now, it's much harder if it's the other, which is where they are delusional and they're really failing badly or disrupting everything and they can't acknowledge it. Once you're locked in with grad students, yes, you can fire an employee, but with a grad student, it's a very tough situation, right? And that's why I encourage everybody because most of our systems, you do a master's and then you transfer. You better be darn sure when you put someone through that it's like getting married, right? It's like 5 or 6 years or who knows how long, right? Never let someone transfer if you have any doubts about it, because you could end up in this situation.
Very good point. And maybe regarding kind of a follow-up on that, how do you ensure that lab ethic is good in your lab? So do you look at the raw data? I think Colin said that he was going through the lab book. So how do you ensure that the graph they're showing you is done the right way and that all the data is correct? How do you kind of control this?
I can start maybe. So I definitely look at the raw data, and if it's something new and a novel finding, I'll have someone else in the lab repeat the experiments. You want to be sure at the end of the day, it's your name on that paper as well. And integrity is number one. So, not because I'm doubting anyone, people make mistakes or, it's little things. So you want to make sure that things are reproducible within your lab before you present it outside. That's the policy, at least for us.
Yeah, we do the same thing, Christina. And the other thing I like to do is frequently, if you're working towards a big finding, there'll be 3 different ways to provide evidence for that finding, right? And so I will frequently have other people doing those other ways. So, someone sees something in a culture dish and maybe someone else will do the in vivo work. And then when they all agree, then you really can, and people in your lab, like in my lab anyway, like that, because they're all nervous about the data too, right? Or they should be if they're not, right? And they want to make sure it's correct. But I just like to say one other thing in that regard. It's really important that we provide environments for the people in our labs that they don't feel pressured to cheat or to misinterpret or whatever you want to call it, right? So if you create an environment where there's no wrong answers, there's only the answer, whatever that is, right? Once you do it correctly, then people aren't going to feel like they have to get a result because that's what you want, or that's because they've been told that's what they need to see to get the paper into a big journal or something.
In terms of being on top of data and reviewing the data of my trainees, I've also found we started an electronic lab notebook in our lab several years ago. And I can literally track the research in real time as it's being done if you do it correctly. And I found this to be very, very helpful. Also, the individual lab notebooks on this electronic platform, you can make it viewable by everyone in the lab if you want. It's very open and transparent way to do science. And I found this has helped out a lot in my own group.
I'm impressed, Colin. It's always been like a dream, but I've never been able to move in that direction. We're so old-fashioned with lab books.
It's I guess to ECIs, it's a good, if you're interested in doing it, it's much easier to start it new than it is to adopt an electronic lab notebook from a big lab.
Colin, which one do you use, just out of curiosity?
Mine is Benchling. It's perfect. So actually, I was charged by one of the administrative duties that I had. I was charged to find an electronic lab notebook for the entire institute, and it had a bunch of stipulations. One being that it was free, and the other being that it needed to be able to have enough data. So Benchling is one of the best available, the free electronic lab notebooks for academics. And I don't work for the company, but would highly, highly recommend it.
Thank you.
You're welcome.
On the perspective of your trainees, Colin, do you is it an additional amount of work for them? Do they have to run their own lab book on paper and then electronic online? So do they have to duplicate their
Some
time
choose
writing?
to have some paper going on, but if you think about it, all of our data is digital. So it really makes sense to have an electronic lab notebook. So if we're at the microscope generating images, back in the day, you would print out the image and then paste it into your lab notebook. And here you just drag and drop the image right into the electronic lab notebook. Also, in terms of keeping things consistent between experiment and experiment, there's standardized protocols that go right into the electronic lab notebook, and you literally can just drag and drop it into your experiment, and then the entire protocol is all written. So and then the new generation, the young students, love this type of technology, in my opinion. So they will easily adapt to it. So I've not had an experience, negative experience using electronic lab notebook yet. But I'm a bit biased. I really wanted to do it. So I guess it's up to you, not telling people how to run their labs.
I think we've been through pretty much all the questions questions that I had and the questions that were on the Q&A. I think we answered to the questions that I still have there in a way or another. I don't want to repeat myself too much and have you talk over and over and over again. So unless you have maybe a closing remark or something that kind of take-home message that you wanna pass on, do you want a last word before I end the session?
Yeah, I'm just going to say, don't forget what made you become a scientist to start with. Sometimes when you're starting, it's intimidating. It can be negative, if your grant's rejected or your paper's rejected. But, hang on to what science is really all about. All the rest of that is just, it's not peripheral, you need it, but it's not the core of it. So, through bad times, that's what I hung on to. And so I hope that helps a bit.
Congratulations to all of you who are starting your new lab. And, I wish you all the luck. Enjoy.
Yes. Congratulations.
Well, thanks a lot to all of you. I think it was a very informative session. A lot of good practical points, a lot of perspective that I think we're all going kind of in the same way. So I guess we can have a good idea that that's at least a good way to do it. There's probably different ways to do it, but I think it's very helpful. At least it was very helpful for me. So I'm pretty sure it's very helpful for all the people that have assisted. Just before I close the session, I have two announcements to make. One is regarding a new opportunity by the Stem Cell Network. So the Stem Cell, I'm just going to read the short paragraph that I have on this new opportunity for ECIs. So the Stem Cell Network is excited to announce the launch of the 2021 Nicolas Meetings Early Career Investigator Speaker Competition. So this competition will provide up to 3 early career investigators with the opportunity to present progress from their research program to the broad stem cell and regenerative medicine research audience at the 2021 TMM meeting in November. In the pre-recorded sessions that will be available on demand by demand viewing by all TMM 21 attendees during and after the conference. So eligible ACIs are invited to submit an abstract outlining their research projects relevant to stem cells and regenerative medicine that will form the basis of their talk. The deadline for abstract is Friday, April 30 at midnight. I don't know if Janice will have maybe something to say about when it's, I guess it's going to be publicized on emails or online also.
Yeah, so everyone on that is a registered participant here is getting sort of a early bird announcement about it. We will include it in cell lines and we'll be promoting it elsewhere, but we wanted to give you all the first notice of this new initiative. Great, thanks. And the last thing I wanted to say is also to remember to the other sessions coming up. So the next session will be next Wednesday, March 31st. Will be the topic will be the art of funding your research. So definitely something also that's very important for ECIs. It will be moderated by Jessica Esseltine. And the panelists will be Karen Singh, Christine Hope, and Michael Rudnicki. So again, a very good panel to talk about the research funding. So once again, let me thank all the panelists for their time and their precious advice. It's been very informative and very nice event. And thank you all for coming and watching and for all your good questions. Janice, anything else to add before we leave? No, thanks to everyone. Thank you to the panelists and thank you to you, Nicholas, for helping us out today. You did a great job and it was very, very well attended by everyone that participated. Thank you. Yeah, welcome.
Thank you all.
Everyone, great to see everyone.
Bye, nice to see you all.
Bye.
Bye-bye.
Bye.